Today I will be discussing a new method for deconvoluting transcriptional regulatory networks from expression data, specifically in bacteria. I'll start with a brief overview of transcriptional regulatory networks, or TRNs, in bacteria, and then I'll introduce the expression compendium we compiled to probe E. coli's TRN. I'll then introduce independent component analysis, or ICA, which is the machine learning method that enables detailed characterization of TRNs. Finally, I'll go over some of the validation experiments and applications of this framework, such as inferring cellular states and environments from transcriptomics data, characterizing mutations acquired in laboratory evolutions, and comparing the transcriptomes across diverse E. coli strains. The Transcriptional Regulatory Network, or TRN, is an information processing unit that takes in metabolite concentrations, pH, signaling molecules, etc., incorporates all of this information, and outputs an optimized gene expression profile for the experimental condition. In bacteria, TRN elements include transcription factors, sigma factors for transcriptional initiation, riboswitches and RNA elements, and RNA polymerase itself. It's the interactions between these elements, the environment, and the genome that comprises the TRN. A common simplification in systems biology is to frame the TRN as just interactions between generic regulators and the genome. Prior work on genome-scale TRNs mostly focused on identifying whether regulators were activators or repressors, and also finding all the genome-wide binding sites of specific regulators. The set of genes that are regulated by a single regulator is called a regulon. Unfortunately, multiple studies have shown that simple regulon membership data is not nearly enough information to predict gene expression profiles. This is in part because regulators can have different quantitative effects on each gene, and that regulator activity is dependent on the specific experimental condition. After all, the main function of the TRN is to respond to changing conditions. We can view the TRN as a function with all of these inputs and outputs. If we have enough diverse outputs, would we be able to actually reverse engineer the TRN? And if we do that correctly, is it possible that we can actually pass a gene expression profile through the TRN and get back the original cellular state? In order to do this, we needed a high-quality compendium of diverse expression profiles. Previously, our lab has published many studies using RNA-seq expression data to investigate functions of transcription factors and properties of E. coli metabolism. In addition, our lab specializes in adaptive laboratory evolution experiments, or ALEs. In an ALE experiment, cells are grown under a constant laboratory condition and acquire mutations that select for increased growth. Until now, the expression profiles from each individual study were analyzed independently using traditional differential expression analysis. However, by combining all of this data together, we can build synergy between the datasets and identify constant underlying patterns across many diverse conditions. Over the last 5 years, our research group has created around 300 datasets. This is about 20% of all publicly available datasets in NCBI-GEO for E. coli. All of this data was created in the same lab using the same protocol, resulting in high-quality, self-consistent data. As you can see here, the median R-squared between replicates is 0.98, and the median R-squared between non-replicates is still 0.7, highlighting the internal consistency that allows us to compare transcriptomics data between experiments. We call this compendium PRECISE, or the Precision RNA Expression Compendium for Independent Signal Extraction. In order to extract the TRN from PRECISE, we turn to an unsupervised machine learning algorithm called independent component analysis, or ICA. ICA was developed to solve the cocktail party problem. Imagine you're in a room with 3 people speaking simultaneously. If you place 3 microphones randomly in this room, each microphone will pick up a different linear combination of each of the source signals. If we apply ICA to these mixed output signals, we can reconstruct the original source signals. We can also estimate the relative volumes of each of the sources on each of the outputs. ICA can achieve this with no inputs other than the recorded mixed signals through properties of statistical independence. If we replace the source signals with transcriptional regulators, and the output signals with the gene expression compendium, we end up with a pretty good approximation of the TRN. In this case, the output mixed signals are gene expression profiles. The signals correspond to transcription factor binding sites, and the signal activities are related to the activity levels of the transcription factors themselves. Mathematically, ICA decomposes the original expression matrix into 2 matrices, one which represents regulator signals and one which represents regulator activities. The S matrix has dimensions of genes by signals, and the A matrix has dimensions of signals by experimental conditions. As I'll demonstrate later, you can think of each signal as a transcriptional regulator. This representation is quite nice because it cleanly separates the condition-invariant network structure that is encoded in the genome as regulator signals from the condition-dependent regulator activities which interact with the environment. Therefore, each expression profile is represented as the sum of each signal scaled by its condition-specific regulator activity. So each column in the S matrix represents the binding sites of a single regulator. Each value in this column represents the effect of the regulator on the corresponding gene. If we plot these values in a histogram, you can see that most of the values are near zero. I'd like to point out that the y-axis here is on a log scale. We can draw a threshold to remove all genes with insignificant coefficients. The set of outlier genes outside of the threshold are called an iModulon because these genes are independently modulated across the entire expression compendium. This is a play on the word regulon, which is the set of all genes known to be regulated by a specific transcriptional regulator. In this example, I've highlighted the multi-regulon, and we can see that it's almost identical to the genes in the iModulon. The S matrix is invariant to the experimental condition as it is entirely encoded in the genome as regulator binding sites. On the other hand, the A matrix represents the activity of a single regulator across all experimental conditions. If we take the row corresponding to the glycerol uptake regulator, GLPR, we can see that its activity is high when glycerol is used as the carbon source and low when any other carbon source is used. To make this interpretation easier, we set a baseline condition, where 0 represents the activity in the baseline condition for any iModulon. This baseline is wild-type E. coli MG1655 grown on glucose M9 minimal media. The overall workflow starts by applying ICA to an expression compendium. We take the S matrix, which encodes the TRN structure, and find outliers, and we call these iModulons. We map these iModulons to the known TRN, specifically from regulon DB, using all interactions, and we name the iModulon if it shares a significant number of genes with a known regulon. iModulons are defined and named solely by their overlap with known regulons. Since we're using all of the interactions from regulon DB regardless of the confidence level, I'm going to call this a silver standard TRN as there may be some inaccuracies due to low confidence interactions. Finally, we can use the regulator activities to gain a deep understanding of the cellular state. So how do regulator signals and activities work together to recreate the expression matrix? I'll illustrate this through the CRA iModulon. CRA is the major regulator of glycolysis in E. coli, and it's activated by fructose 1-phosphate, as you can see here. So this is a network view of glycolysis with glucose at the top and citric acid cycle at the bottom. We found one iModulon that was enriched in genes regulated by CRA. If we map these genes onto glycolysis, we can see there's a pretty good overlap. All of the genes in blue have positive iModulon coefficients, and all the genes in red have negative iModulon gene coefficients. The genes in red are associated with gluconeogenesis, whereas the genes in blue are mostly associated with glycolysis. We can think of the iModulon activities as a big volume knob on the expression level of the iModulon genes. So when we grow it on glucose, this is our baseline condition, so the activity is 0. All of their activities are relative to this condition. When we grow the cells on fructose, CRA is activated and all the genes in blue are upregulated and all the genes in red are downregulated. This corresponds to increasing expression of glycolysis genes and decreased expression of gluconeogenic genes. However, if we grow E. coli on a poor carbon source such as pyruvate, the genes in red are overexpressed and the genes in blue are underexpressed. So now I'll talk about the 92 iModulons we get when we apply ICA to our precise expression compendium for E. coli. So we split the iModulons into different types. Most iModulons correspond to a single transcription factor. As you can see here, all of the genes in the CDAR iModulon, well, all of them except for one, are regulated by CDAR. We also see enrichments in sigma factors, so here we see the FLIA sigma factor iModulon. We can also get iModulons that are enriched in multiple regulators. For example, E. coli senses zinc using two regulators, ZNTR and ZUR. Both of these regulators are captured by a single iModulon, since they're actually capturing the same initial signal, the zinc concentrations. Finally, we get a lot of genomic iModulons. These are usually due to engineering changes, for example, a gene knockout. In this case, this iModulon has THRA as the dominant gene in the iModulon, and it's only active when we knock out THRA in the expression compendium. For the remainder of this talk, we're going to focus on regulatory iModulons. To assess the confidence we have in these regulatory iModulons, we're going to use precision, which is the number of genes in the iModulon and in the regulon divided by the total number of genes in the iModulon. We're going to ignore extra genes in the regulon because sometimes regulons can be split between two iModulons. The average precision of our iModulons is about 80%. We chose two iModulons whose precision was slightly lower to validate whether the predicted genes were actually part of the regulon or not. So the METJ transcription factor regulates methionine biosynthesis. The iModulon contains 6 extra genes that are not originally reported in the METJ regulon. We performed ChIP-EXO to find the genome-wide binding sites of METJ, and we were able to confirm every single gene in our iModulon. In addition, we did not find binding sites upstream of the 2 genes that were reported to be in the regulon, but not found in our iModulon. The other transcription factor we wanted to validate was cis-B. The cis-B regulon was split into 2 iModulons. One of them was just regulated by cis-B, and one of them was co-regulated by both cis-B and CBL. When we performed ChIP-EXO on cis-B, we were able to validate every single gene in both iModulons except for one. In addition, we only found 2 genes that were in the regulon and not in either iModulon that had binding sites upstream of them. So now that we have reasonable confidence in the iModulons, we can look at what biological phenomena can be recapitulated by this framework. For example, here I've shown the xyl-R iModulon. Many of the genes here are known to be regulated by xyl-R, but many of the genes have no evidence of xyl-R regulation. When we investigate the activities of the xyl-R iModulon, we see that the activity is very high when xylose is the primary carbon source, and it's near 0 when it's any other carbon source, similar to the glycerol iModulon described earlier. Many of the genes in the xyl-R iModulon are Y genes, or uncharacterized genes in E. coli. The only characterization for the YIA genes are that they're putative xylosidases, and the YIC genes is that they're putative transporters, but unknown what they transport. The YAG genes have no annotations whatsoever. Since these genes are co-modulated with other xylose uptake genes, we can get a sense of what these genes' true functions really are. Another example iModulon is the EVGA iModulon. Again, you can see many of the genes are regulated by EVGA, hence why we named it EVGA. We can also look at the activities, and EVGA is known to be active under low pH and osmotic stress. We see this perfectly recapitulated where all of the expression profiles with low pH or osmotic stress have high iModulon activities, whereas every other experimental condition have low iModulon activities near 0. So this brings us to our first application, which is using transcriptomics data to infer cellular states and environments. One iModulon is enriched in genes regulated by the RPOS general stress response sigma factor. This iModulon has very high activities under stressful conditions, such as osmotic and oxidative stress and various starvation conditions. However, in our evolved laboratory evolution endpoints, we see that the RPOS iModulon activity is very low. Specifically, we found that mutations in RPOB and RPOC, RNA polymerase subunits in E. coli, had very low activities. Another iModulon we found was enriched in genes related to translation, for example, ribosomal subunits. The activity levels of this iModulon were highly correlated with the growth rates measured under these experimental conditions. When we plot the RPOS iModulon activities against the translation iModulon activities, we see a clear trade-off between fear and greed. When cells are experiencing stress, they shift to the bottom right corner of this plot, the fear side. However, when cells are unstressed, they grow much faster, which is the top left point in this plot. We have even been able to identify 2 point mutations in RNA polymerase that shift cells up this line. We can also use iModulons to probe the internal state of the cell. The ferric uptake regulator, or FER, is inactive when there's no iron present. When iron is present in the media, it binds to FER, which activates it, and FER then represses a lot of iron uptake genes. The FER regulon is split into FER1 and FER2 iModulons. We'll focus on the FER1 iModulon for now. When E. coli is starved of iron, we can see that the iModulon activity of FER1 is high. This is consistent with the apoform of fur. However, when we add iron to the media, the genes are repressed due to the active form of fur. In one experiment, we knocked out 2 genes that convert charismate to isocharismate. Isocharismate is the precursor to both menaquinone and enterobactin. Enterobactin is the main way that E. coli K12MG1655 uptakes iron from its environment. When we knock this out, we see that this double knockout has iron starvation characteristics in the fur1 iModulon, meaning that it's unable to uptake iron. However, when we evolve it in our adaptive laboratory evolution machine, we find a pseudogene that encodes an elemental iron transporter is actually repaired. This transporter now uptakes even more iron than before, exhibiting a very low fur1 iModulon activity. This means that the genes in the fur iModulon are very lowly expressed, as what would be expected when fur is active and bound by iron. So the iModulon activities offer a snapshot of all of the molecules and signals that the cell is able to sense through its TRN. This includes media components, as displayed by fur, stressors and starvation, as shown by the RPOS iModulon, and other environmental factors, as we discussed with EVGA. This simplifies diverse transcriptional changes in complex environments to simple changes in regulator activities. The next application I'll talk about is characterizing mutations acquired during adaptive laboratory evolution, or ALE experiments. In one ALE experiment, there is a truncation in the transcription factor MALT. If we look at the MALT iModulon, we see that its activity is extremely low. This shows that MALT, which is an activator, is no longer able to activate or upregulate its target genes. In another example, there are 2 instances of mutations in CSQR in our database. One mutation results in the truncation of the transcription factor, and another mutation is a SNP. Both mutations increase the iModulon activity of the regulated genes. Since CSQR is a carbon source uptake repressor, we can see that both of these mutations deactivate CSQR and prevent it from binding and repressing its target genes. Another example is in RBSR. When we grow E. coli in ribose M9 minimal media, where ribose is the primary carbon source, we see that the activity is high. In one of our ALE endpoints, we actually see that RBSR is truncated. This has the same effect as growing it on ribose media, which de-represses the ribose uptake operon. We can also use iModulons to characterize new regulons. We found one iModulon with high activities only in strains with mutations in YGBI. When we looked at the genes in the iModulon, we saw that all of the genes were actually divergently transcribed from YGBI itself. YGBI is a putative transcription factor, but no information was known about it. Given that these mutations allowed the cell to grow on a novel carbon source, we predicted that YGBI was a repressor of all the downstream genes in this operon. The various mutations in YGBI deactivated the transcription factor, allowing constitutive expression of the YGB genes. Another iModulon had a specific ALE endpoint where the activity was extremely high. When we look at the genes in this iModulon, we see ProQ and NC have negative iModulon coefficients, and GLSB, YNEG, and SAD have high iModulon coefficients. These 3 genes with positive iModulon coefficients are divergently transcribed from YNEJ, yet another uncharacterized putative transcription factor. YNEJ was mutated in this ALE endpoint, and so we predict that it regulates these 3 genes. ProQ and NC are actually deleted in this strain, explaining their negative iModulon gene weights. So this shows that iModulons can connect acquired mutations to downstream physiological effects. This allows us to connect the genotype to the phenotype. In addition, the ALE experiments themselves provide context to mutated regulators. For example, with YGBI, we were able to predict that YGBI regulated carbon uptake. In these ALE studies, the original question that was asked had nothing to do with the transcription factors. However, ICA revealed answers to questions we didn't even ask. This shows that compiling a large expression compendium for any organism can give us a shotgun approach to characterizing the TRNs of new organisms. The final application of ICA is in comparing the transcriptomes of closely and distantly related E. coli strains. We found one iModulon that discriminates between 2 closely related strains in E. coli. Our compendium contains E. coli MG1655 and BW25113. These two strains only diverge by 30 mutations. One iModulon was able to clearly differentiate between these two strains. When we look at the genes that are in this iModulon, all of the genes with the negative coefficients down here are either deleted or truncated in the BW strain. So this negative gene coefficient multiplied by the positive activity in BW shows that there is a net negative expression level compared to the wild-type MG1655 strain. On the other hand, the genes with the positive iModulon coefficients all have mutations we hypothesize increase their expression levels. For example, there's an IS30 insertion element added in MHPC in the BW strain that's not there in the MG1655 strain. IS30 is known to have an internal promoter, and this seems to cause constitutive expression of the MHP operon. There's also a SNP at the transcription start site of the TAB-A YJGL operon. This seems to also increase the expression of these two genes in the BW strain. Finally, there's a SNP in FAB-R, which is a regulator of FAB-A and FAB-B, which seems to reduce its ability to repress these two genes. And there's also a mutation in the phenylalanine tRNA. This participates in tRNA-mediated attenuation of Phe-A. Therefore, this iModulon represents a general transcriptomic transformation between the MG1655 strain and a generic BW25113 strain. We then expanded this to look at 8 different strains of E. coli, grown under identical conditions. 3 of these strains diverged from a common ancestor, the K12 strains. These ones had very similar iModulon activities. In addition, we can see pathogenic strains clustered together in their iModulon activities. In particular, we looked at the RPOS iModulon, which had the most variability in its iModulon activities. When we look at the RPOS protein sequence in all these strains, we can see 3 alleles. One has glutamine, one has glutamate, and one has a stop codon. The inferred iModulon activities for RPOS are lowest in the truncated version of RPOS, and are highest in the allele with glutamine in position 33 of the protein. The switch from glutamine to glutamate seems to reduce the iModulon activity, but not nearly as much as a truncation would. Thus, we can see that iModulons can differentiate between very closely related strains. We can use this to identify the transcriptomic differences between resistant and susceptible pathogenic strains. In addition, we can use a representative TRN built from the expression profiles from a single strain to estimate the regulator activities of all other strains. This allows us to infer regulator activities for not just laboratory-evolved strains, but also clinical strains. So in conclusion, ICA is a very valuable tool to understand bacterial TRNs. The iModulon activities represent any signal that can be measured by the cell. This allows us to infer the intracellular state of an organism while in complex environmental conditions. iModulons also enable characterization of unknown genes and regulators, as shown by the YGBI and Y and EJ iModulons. Gathering a detailed TRN from a single strain can actually reveal properties across multiple strains, including clinical isolates. And finally, these results show that reanalysis of old data can have powerful effects and can answer questions that were unasked by the original study themselves. I would like to thank Dr. Bernard Paulson and Dr. Adam Feist for their support and contributions to this presentation. In addition, I would like to thank the rest of the Systems Biology Research Group, specifically Sagat, Yi, Richard, Ying, Julia, and Amitesh, who have contributed many of the datasets analyzed in this presentation. We are funded by the Novo Nordisk Foundation Center for Biosustainability and grants from the NIH. A preprint of this manuscript is available on bioRxiv at the link provided here.